Dear students, we will continue the previous lecture, Schistosomiasis. The outlines of this lecture are we will discuss Schistosomiasis. We already discussed in the previous lecture the introduction and epidemiology, etc. Today we will discuss life cycle, pathogenesis, treatment, preventions, etc. Lecture outcomes. So after watching and listening to this lecture, student will know about schistosomiasis. So in continuation to the previous lecture, we were discussing the release of the egg and activation of the meracidium. So the first indication of hatching is activation of cilia on the meracidium. So it is the egg is hatched if the activation of cilia occur on the meracidium. So it means that it is hatched. So ciliary activity increase until the meracidium is uh, veritable spinning ball. It means that the ciliary activity will increase, and as the ciliary activity increases, the meracidium will look truly variable, mean it truly will look like a spinning ball. Then suddenly an osmotically induced vent open on the outside of the egg and the meracidium emerges. I will show you the figure in the next slide, 16.10. So osmotically induced vent of the egg gate will suddenly open on the side of the egg or at one side, and the meracidium will come out of this egg. The meracidium usually contract a few times, completely clearing the shell. So with that contraction and the ciliary movements, it will completely clear the shell, after which it rapidly swims away, and then it will swim into the water in search of the next host. However, some eggs do not hatch no matter how active the meracidium becomes, and others hatch before the larva becomes activated. Oh, this is the egg. You can see the spine at one stage, so it's the indication of the egg, the Schistosoma mansoni egg. So this is the egg, and this is the meracidium, and because of the opening at one side, the meracidium, with the help of contraction and the ciliary movement, will come out of this shell and will swim into the water. Uh, I think we already have discussed these structures in detail, but the diagram is here, so we will explain the diagram. This is actually the male schistosome, and this is the female schistosome. We have already discussed in the previous lecture that there is a gynecophoral canal on the ventral side, a groove which is known as gynecophoral canal in the males, where the female normally live. This is the oral sucker, and this is the ventral sucker. This is the esophagus. Then this is the genital pore through which the sperm are releases. These are the testes, and this is the cecum, and this is the gynecophoral canal, and this is the intestine of the male. Then if you look at the female, you can see there is the oral sucker, and this is the ventral sucker, and then there is the genital pore. This is the uterus, and there is this Meles gland, O type, and this is the ori, and this is the vitelline duct. This is the intestine, and in the last you can see the vital area. So this is the general morphology and anatomy of the schistosome, male and female worm. Meracidia are typical and swim cautiously mean continuously during their short life. Because they have very short life, then they have to like enter into the next one. So if hatching from an old egg, a Meracidium will live only 1 to 2 hours, and optimal condition it will survive for 5 to 6 hours. So it has a very short life after its hatching, and it must reach the next host for continuation of its life. So if in a Meracidium it like hatch from a very old egg, so then Meracidium will live only for a short period of time, 1 or 2 hours. Or in optimal condition, if the conditions are very optimum, so then it will survive. It may survive for 5 to 6 hours maximum. So although Meracidia of Schistosomes do not have eye spots, they apparently have photoreceptors, and they are positively phototropic, meaning they swim toward light. They don't have like eye spots, but they have photoreceptors. When Meracidia enter the vicinity of a snail host, because the next host after it emerges from the egg, so for the Meracidia, the next or the immediate host is a snail. So it will look for a snail to enter into its body and completes its next phase of life. Oh, they are like stimulated to swim more rapidly and change direction much more frequently, thus increasing their chances of encountering the host. So once they are in a vicinity, mean in an area, so once they are in the snail's area, which is its next host, they are stimulated to swim more rapidly, and they change their direction more frequently, meaning again and again, in search of a host, and to enter to its host body quickly or as soon as possible. So following are the most important snail species in which they complete their life cycle, and figure 16.7. So here in this picture is the complete life cycle of Schistosoma mansoni. There are three main species of important. There are many other species, but there are three main important species of Schistosoma, which is Mansoni, Hematobium, and Japonicum. For these three, for all these three, the snail species which are the intermediate host for them are different. Like for example, for the Schistosoma mansoni, the species of snail is Biamphilaria. This is the Biamphilaria snail species. For Schistosoma hematobium, the snail species is Bulinus. This is Bulinus snail species. And for that Schistosoma japonicum, Oncomelina is the right species. So this is the snail species Oncomelina for japonicum, Bulinus for hematobium, and Biamphilaria for mansoni. Okay, so you can see in the structure, this A is embryonated egg is passed in feces. B is Meracidium hatches spontaneously and penetrates Biamphilaria. So it will like enter into this snail species, which is the next host, the intermediate host. C, as a two Sporocyst generation develop in snail. D is Circadia larva, which leaves the snail and penetrate in the skin of the definite host. So the next phase is the entry into the definite host, which is human being. So now it will enter into the human being. So the adult schistosomes mate in portal venules of intestine. This is the actual adult schistosomes. This is the male schistosome in the Gynecophoral Canal, where the female lives. So once they like produce the eggs, and those eggs they enter into the colon, and then they pass out, and the fecal material and the feces outside into the water. So as once they reach the water, the embryonated egg it hatches, and the Meracidium will come out. So here, like the egg hatches in water, and the meracidium larva will come out from the egg casing. So then in the next phase, this meracidium, it will enter into or penetrate into the skin of the Biamphilaria species of snail, and it will complete this phase here, and it will like grow inside the snail into a next stage, which is known as Circadia larva. Now when the stage is complete, Forcocercus, because of this because of this fork tail, this is also known as Forcocercus larva. So this Circadia larva, when it is like complete, it will come out from this Biamphilaria species and will enter or release into the water, where it will find its way and it will bore into the skin. Once it come in contact to the next host, the definitive host, which is a human being, and it will like bore into the skin through boring into the skin, and it will reach to the liver and mesenteries, or the alimentary canal. In the previous slide, we discussed uh, different species of Schistosoma hematobium with respect to their snail host. There are here are some other snail species apart from we discussed in the previous slide, which also act as an intermediate host for Schistosoma hematobium and Mansoni and japonicum. So for Schistosoma hematobium, several species of Bulinus are acting as intermediate host here, also of Isopsis, and possibly also the Planovirus are also acting as intermediate host for Schistosoma hematobium. In case of Schistosoma mansoni, Biamphilaria is the common or the main intermediate host. Biamphilaria alexandria in northern Africa, in Saudi Arabia, and in Yemen. Then there is Biamphilaria sodaniga, then there is Biamphilaria rupeli, then there is Biamphilaria pfefferi, and other in the genus in other parts of Africa. And then there is Biamphilaria gibrata in the Western Hemisphere, and Tropicobrus sentimentalis in Brazil. So these are like different snail species for Schistosoma and mansoni in different areas of the world. So once again, Biamphilaria alexandra in the northern Africa, Saudi Arabia, Yemen. This sodanica, Biamphilaria sodanica, rupeli, and pfefferi, and others. There are many others in the genus and other parts of the world. So apart from these three, there are many others which are also included in this genus, and they are present in either different parts of Africa. Then there is Biamphilaria gibrata in the Western Hemisphere, and then there is Tropicobrus sentimentalis in Brazil. Then for Schistosoma japonicum, there are several species of Oncomelina. In this figure and in the coming figure, we will discuss different sizes of different eggs of different species of Schistosomes. So eggs of Schistosome flukes from A to H, egg sizes from Luker, the scientist, or from his book, and the sizes are in micrometers. So keep in mind, when we will discuss the different sizes, you have to keep in mind that the sizes are given in micrometers. So the first one in figure A, the eggs belong to Schistosoma mansoni, which size is 142 cross 60 micron meter. And in figure B, or in the diagram or the picture B, you can see Schistosoma intercalatum, which size is 175 cross 62 micron meter. And C, you can see the Schistosoma bovis, which is 202 cross 58 micron meter. And D, you can see Schistosoma rodeni, which size is 149 cross 55 micron meter. And E, you can see Schistosoma methei, which size is 173 cross 53 micron meter. And in F, you can see this Schistosoma japonicum, which size is 81 cross 63. And figure G, you can see the Schistosoma jautii, which size is 91 cross 68 micron meter. And here in H, you can see the Schistosoma hematobium. It is the egg of Schistosoma hematobium, which is 144 cross 58 micron meter in size. And here in this cross section is a cross section of the skin, and here you can see the Circadia, which entered or penetrated into the skin of a scientist, and it's moving toward its point now. So we were like discussing the life cycle. So when the meracidium, when it come out of its shell, and the next target is the Biomphilaria larva, so it will enter into the body of the snail for the next phase. And after penetration of a snail, a meracidium will shed its epithelium and begin development into a mother sporocyst, usually near its point of entrance. So near the point of entrance, this meracidium stage, now it will shed its epithelium, its skin, and it will develop into a mother sporocyst, and it will very near to the point of entrance. So after about 2 weeks, the mother sporocyst, which has 4 protonephridia, give birth to daughter sporocyst, which usually migrate to other organs of the snail if there is room. Of course, if there is room for others. So this mother sporocyst, which has with 4 protonephridia, the excretory organs, of course, you know that, it will now start giving birth to daughter sporocysts if there are space, and it will migrate to other organs of the snail. So now the mother sporocyst continues continuously, it will continue to produce a daughter sporocyst for up to 6 or 7 weeks. And keep in mind that there is no radial stage in Schistosoma life cycle. So the Circadia larva, as I showed you, the Circaceous, because of the bifurcated, or this is the fork shape tail, start to emerge from the daughter sporocyst, and the snail host about 4 weeks after initial penetration by the meracidium. So it was 4 weeks ago when the meracidium it penetrated to the body of the snail, where it developed into mother sporocyst. It started producing Circadia larva, and that Circadia larva, or the Circadia larva, then they start emerging after 4th week of its initial infection. Now the Circadia larva will start coming out from this snail species. This is the Circus Circadia larva, because of its forked tail. So this Circadia have a body of 175 micron meter to 240 micron meter long, and its body is 55 micron to 100 micron meter wide. Its tail is 175 micron to 250 micron long, and 35 micron to 50 micron wide, bearing a pair of furky 60 micron to 100 micron long, that bifurcated tail. It is forky. In oral, sucker is absent, being replaced by a head organ composed of penetration gland, and the ventral sucker is small and covered with minute spines. 4 types of glands open through bundle of ducts at the interior margin of the head. This is the mother sporocyst, and when this mother sporocyst is ruptured, these Circadia are released, having these Furcocercous tail. That's the Furcocercous Circadia. Because of this fork-shaped tail, it is known as Furcocercous Circadia. So when the mother sporocyst it rupture, these are ready to release, and then it will penetrate, like enter into the next stage, will come out of the snail body and will in the water, and will now reach to its next host. So these are the Circadia, and like you can see the head of these Circadia. This is the head, and this is the body, the main body, and this forked tail, having these different ducts, glands, and spines, sucker, and a penetration gland into for penetration into the next host. So now, after completing it a stage of its life cycle inside the snail, now it will emerge from the snail, and now it will start preparation for entry into the next host, which is a human being, and there is no second intermediate host in its life cycle. So now Circadia alternately swim to the water surface and slowly sink toward the bottom, continuing to live this way for 1 to 3 days. And after that, if this Circadia come in contact with the skin of a prospective or a suitable host, such as a human being, so they attach and they will creep and slowly will creep on the surface of its host, the human being, seeking for a suitable place to penetrate a soft or a cracked part, where it can easily penetrate into the skin of its definitely host, the human being. How they find their host? So they are actually attracted to the secretion of the skin, and they have a strong sensation or a strong positive response for the amino acid arginine. So once they are like at the skin, when they reach to the skin of the host, and they are attracted by the amino acid arginine. So once they are like once they reach to the skin of the human being, they by themselves, the Circadia by themselves, will start secreting arginine from its some of its gland, acetabular. From the post acetabular gland, they will start secretion arginine by themselves. Why? Because to like attract other Circadia, if they are in the neighborhood, if they are in the near vicinity, so they will be attracted to that arginine, and they will combine like penetrate in the skin of the host. So the Circadia larva, they require half an hour or less time to completely penetrate to the epidermis, and they can disappear through the surface in 10 to 30 seconds. And I have shown you in the figure 16.8 where a Circadia was penetrated into the skin of a peristologist, and it was inside there, and it was like trying to get to its point. Now penetration is like accompanied by a vigorous wiggling movement, like wiggling movement to bore easily into the skin and to reach its destination. So together with the secretion of the products from the head organ. Now this penetration is it is accompanied by the vigorous wiggling movement, and also from the head organ there are secretion, the products which helps in penetration. The tail will drop off in the process. During the process of penetration, the tail will drop out. These worms become somewhat very smaller when the penetration gland are emptied, when their contents are released to help in the penetration, and the worm will become now very smaller, because now the contents are, the tail is lost, and the contents in the head for the penetration is also like secreted. So now the worm will become very smaller in size, and can easily like move or carried out in the veins. So within 24 hours, the schistosomules now enter the peripheral circulation, that will enter into the circulation, and where they are swept off to the heart. So sometime, some of the schistosomules may migrate through lymphatics. So not only to the circulation, they may enter into the lymphatics to the thoracic duct, and from there to the subclavian vein, and then to the heart. So like leaving the right side of the heart, the small worms will wriggle their way through pulmonary capillaries to gain access to the left heart, and so then to the systemic circulation. And migration through the lung capillaries is evidently a major obstacle, and up to 70% be eliminated there. It appears that only schistosomes that enter the mesenteric arteries and then make its way to the intestinal capillary bed transverse, now break through the intestinal capillary bed, and then reach to the liver by the hepatic portal vein system, hepatic portal system, can continue to grow, and these are mostly like eliminated by the host immune system. After undergoing a period of about 3 weeks of development in the liver sinusoids, the young worms now they will pair up. Pair up mean that the female will go inside the gynecophoral canal, and when they pair up, then they migrate to the gut or bladder wall, and then they start beginning producing eggs there. The entire prepatent period is about 5 to 8 weeks. Adult schistosomes may live 20 to 30 years, and the unpaired female worms do not become sexually mature and have the appearance of a starving worm. Their esophageal musculature is very weak and thin. They produce little of at least some digestive enzyme, and they ingest about 1/4 as many erythrocytes as the paired female. So, like if they are not paired with the male, so that they cannot produce eggs, and they cannot continue their life, and they will they will appear like a starving one. So a growth stimulating function may result from the muscular action of the clasping male, because the male musculature is very strong, and it clasps the female, which helps the immature female pump blood into her intestine, because the musculature of its esophagus is very weak. So it is like helped by the male for like pushing or pumping the blood from the sucker to the esophagus and then to the intestine. Surprisingly, the normal development of schistosomes require like cues mean help from their host immune system. So without the facilitation of the host immune system, this schistosome normal development is not possible. And how it happens, you can see it. TNF stimulates egg production, and growth is impaired in IL-7 deficient mice. So if IL-7 is not present, the growth will be impaired, and the TNF stimulates the egg production of the schistosomes. Parasite growth is stimulated by IL-7 and thyroxine, and CD4 plus lymphocytes are an important part of the immune signals that are recognized by the parasites. So it's amazing that the host immune system help or facilitate the normal growth or the normal life of the worms. Like you can see, the egg production is stimulated by the TNF, Parasite growth is stimulated by the IL-7 and the thyroxine, and CD4 lymphocytes are very important, part of the immune signals that are recognized by the parasite. These are the references, and thank you so much for watching and listening. See you in the next lecture.